Hi everyone, welcome to all. In this video, we'll be learning about third program of machine learning lab. So in this program, we are using principal component analysis or a PCA to reduce the number of features in a dataset. We will apply PCA to the famous iris dataset, which is one of the most famous and basic datasets used in a machine learning, which originally has the four features, that is sepal length, sepal width, petal length and petal width. Our goal is to reduce these four features down to just two new features while still keeping most of the important information. This process is called dimensionality reduction. So why do we do this? To make the data easier to visualize, to reduce the noise and redundancy and improve performance in a further analysis. Okay, let's start with the code. As we all know, NumPy, this is a library, helps us handle numerical operations. And pandas, which is useful for working with our tabular data. Then here, we are loading the famous iris dataset using a scikit-learn. It contains data about 3 types of iris flowers with 4 features. We import a PCA or a principal component analysis, which is the technique we will use to reduce the number of features from 4 to 2 while keeping the important patterns in the data. Matplotlib, which helps us to plot graphs and visualizations. Yes. See, this line is used in a Jupyter notebooks to make a plots show up directly below the code cells. It's not required if you are running this in regular Python script. Yes. Then in the next line, see, this line loads the full iris dataset into a variable named iris. Yes, it includes both the measurements and the class labels for each flower sample. So here we extract just the feature values. This is a two-dimensional array containing the 4 measurements, that is sepal length, sepal width, petal length and petal width. This line extracts the actual flower species labels for each row. And this line gives the human readable names for those species. Then in the next line, here we are converting the raw data into a pandas data frame, which is like a table, yes, similar to an Excel sheet. So we can also assign a proper column names using iris.feature underscore names, so the table is easy to understand. And this function prints the data frame. This dot shaper tells us how many rows and how many columns we have, and then dot info. As we have discussed in the previous program, it gives the summary of the dataset. Dot head function shows first few rows of the dataset, and dot tail function shows last few rows of the dataset. Next is, see here we are checking for any missing values in the dataset. Is the null function returns true for missing values, and sum function counts how many are in each column. Then value underscore counts. See, this shows how many times each unique row appears in the dataset. It helps us spot if any rows are repeated or duplicated. Then info function provides a summary of the dataset. Then next is describe function. This generates descriptive statistics for each numerical column like mean, standard deviation, min, max and quartiles. We can see in the table. Then in the next step, we are applying principal component analysis to reduce our data from 4 dimensions to just 2. Yes. So here we are creating a PCA object. These are the new axes that capture the most important patterns in the data. And then in the next line, yes, see, this line actually performs the PCA transformation. It fits the model to our 4-dimension data to transforms it into 2D. Then see, we convert the 2-dimension data into a pandas data frame so we can work with it easily. So we name the columns principal component 1 and component 2 to indicate these are new features created by PCA. Yes, And we also had a new column for the original class labels, so we know which species each data point belongs to. Then we set the size of the plot to 8 inches wide and 6 inches tall. Then in the next, we prepare a list of colors to represent the 3 flower classes: red, green and blue. Yes, this is the for loop which goes through each flower type, picks out its PCA, reduced X and Y values, and plots them using a unique color and name. So we can see how the flower species are grouped in 2D space. And label is a class number like 0, 1 or 2, and I is just the indexer we use to pick a color for that flower type. And this selects only rows for the current flower type, and principal component 1 and 2 get the x-axis values and y-axis values from the PCA results. And this sets the label name, and it is for color. This is the title, we can see here. This labels the x-axis with the principal component 1, the new first dimension created by the PCA, and this is second most important dimension from the PCA. And this legend method displays a legend so we know which color represents which flower species. Then this adds a grid to make it easier to read the plot and compare values visually. Yes, so finally it displays the complete scatter plot on the screen. Yes, and this is our output. See, in the output, setosa is nothing but red dots, which is clearly separated from the others. Then versicolor and virginica, those are nothing but green and blue color are closer together, yes, and overlap a bit. This tells us they are somewhat similar, so the model may find it a bit harder to distinguish between them. So the spread of points shows us how the flowers are clustered based on their measurements. Yes, even after reducing the dimensions. So thanks for watching. If you found this video helpful, don't forget to like, share and subscribe for more videos on this, and see you in the next video.